Buenos días a todos nuevamente. Gracias por estar aquí en nuestro segundo día de nuestro simposio. Vamos a iniciar con un invitado internacional, del cual tengo el placer de presentarlo a ustedes. Entonces vamos a cambiar al inglés para hacer la presentación. Pero cualquier pregunta que tengan, vamos a hacer el esfuerzo de hacer las traducciones en inglés, spanglish o como se pueda, pero lo vamos a lograr. Entonces vamos a iniciar con la presentación de el doctor. Voy a cambiar al inglés y iniciamos formalmente nuestro trabajo el día de hoy. Creo que todos me escuchan bien, ¿verdad?
Sí.
Perfecto, muchas gracias. Por cierto, perdón, una aclaración. La doctora Riquelme, yo soy Jimena Carrillo, la doctora Riquelme nos va a apoyar con la segunda parte de esta sesión de la mañana y yo voy a presentar al doctor internacional, a nuestro invitado internacional. Good morning, everyone. Welcome back to our symposium. Today, we will open our second day with an extraordinary international invited speaker. I'm pleased to introduce Professor Avi Schroeder from the Wolfson Department of Chemical Engineering at Technion in Israel Institute of Technology. Dr. Schroeder received his chemical engineering undergrad, master's, and PhD degrees from the Ben Gurion University. Then he did a postdoc at the MIT, but since 2012, he has been the head of his own lab, the Targeted Drug Delivery and Personalized Medicine Group in Technion. He has close to 7,000 citations and an H index of 34, according to Google Scholar, for many, many papers and patents. I counted yesterday like 27 patents and close to 80 articles. He is an expert on the design of smart nanocarriers, or nanoparticles, for the on-site delivery of drugs. His group current research topics include personalized cancer treatments, where a nanoparticle-based technology helps predicting the therapeutic potency of drugs, synthetic cells for producing therapeutic proteins inside the body, targeting tumor microenvironment to treat and target the supportive environment of cancer cells, proteolytic protein delivery, better known as nanoparticle surgical blades, targeting metastasis with liposomal nanoparticles, and last but not least, agricultural nanotechnology. So that's the main reason why he is here today, the use of drug delivery systems in agriculture. So I don't want to steal Dr. Schroeder time, so this is just a summary of his career. But I want to encourage everyone, especially students, to Google him and watch his many videos on YouTube or visit his page websites. So please, Doctor, go ahead and welcome. And thank you very much for accepting our invitation.
Thank you very much. It's a great privilege being here together with you, and please, if you have questions, just bring them up, and I'll try to answer. And what I'd like to talk about today is really that intersection between nanotechnology and agriculture. And as you can see, we're different regions of the world, but close at heart. And I hope the next meeting can be in Mexico or in Israel, in Jerusalem or Tel Aviv or Haifa. I've never been to Mexico before, so I look forward to visiting there at some point. And this is where I'm located. You can see right next to the Mediterranean Sea in Israel. The Technion, Israel Institute of Technology, is one of Israel's universities. It resides in the city of Haifa. The city of Haifa is about 1 hour by car north of Tel Aviv, and an hour and 20 minutes by car northwest of Jerusalem. So it's like a triangle of the three big cities for people who know the geography. And you can see we oversee the Mediterranean Sea. And this is Haifa, the Baha'i Gardens in Haifa. Of course, Israel has many attractions. Jerusalem, the heart of the city, of the country. And Tel Aviv, which oversees, is a very lively city, in Eilat, in the south of Israel, for people who may like scuba diving. And I want to encourage every person to come and visit us. We'd be very happy to host you. So we'll be talking about nanotechnology and agriculture. And when we look at this field, actually, just a couple years ago, has already been highlighted as one of the future areas within agro, in a beautiful set of articles in Nature Nanotechnology. And I want to encourage any person that's interested in this field really to take a look at this collection of articles, which really highlights the potential of where nanotechnology can help in agricultural use. And I'll try to show some of the applications of nanotechnology in agriculture, and really how I reached this field from a totally different field of nanotechnology and medicine. So I'll talk a bit about my path reaching there. I just want to make sure, can you hear me well?
Yes. Yes, absolutely.
Okay, perfect. Thank you. So my group at Technion is composed mainly of engineering students, chemical engineers, biotechnology and food engineers, biomedical engineers, and biologists and chemists. We have also some physicians, and this thank to our funding agencies. And really what brought me into the field of nanotechnology and medicine is this paper you can see in front of you, which was looking at how nanoparticles distribute inside the body of cancer patients. So in this paper, they were looking at different cancer patients. You can notice on the left-hand side of the screen is a patient with a tumor at the base of the tongue, a patient with breast cancer and metastasis in the local lymph node, and a patient with lung cancer and metastasis in the spine and in the bone marrow. And while these patients all have different types of tumors, different types of cancer, even though they had nanoparticles that were injected into their arm, the vein of their arm, the nanoparticles circulated in the blood and actually targeted the different sites of the tumors. And for me, that was amazing, that you can use nanotechnology in order to direct the medicine inside the body exactly to where the disease resides. And that's what really brought me into this field. The reason this happens in medicine is that the blood vessels we have in healthy tissue are different than the blood vessels we have in diseased tissue. More specifically, in healthy tissue, the blood vessels are a little bit like a hose, a pipe that has no holes in in its surface. Contrarily, in sites of cancer, the blood vessels actually have small holes, nano holes in the wall of the blood vessel, which allow nanoparticles to extravasate from the circulation. This would be the blood flow. These would be the nanoparticles, and actually accumulate in the diseased tissue. And this occurs due to a process called angiogenesis, the rapid formation of blood vessels inside the tumors. These blood vessels are different than healthy blood vessels. They actually have small nano holes inside them. Nano holes, about 200, 300, 400 nanometers in diameter is the hole in the endothelial wall. So nanoparticles have to be smaller than that in order to exit the circulation and enter into the diseased tissue. We know today that usually the goal, the size is about 100 nanometers in diameter. That allows the accumulation of the nanoparticles into the tumor tissue. So when I think of a nanoparticle, I always imagine like a tennis ball, and it's composed of different materials. We use mainly lipids. For our agricultural projects, we use lipids that are sourced from plants, such as soybean. And these lipids actually form vesicles in which we can load different types of active agents. If we talk about medicine, nanoparticle, which would be, for example, around 100 nanometers in diameter, will have a medicine inside, and then it will circulate in the blood and really target the diseased tissue through those leaky blood vessels. You can decorate the surface of the nanoparticles with different molecules in order to direct them to one tissue or another. And you can play with many, many different parameters. But the most important is that a particle that reaches a target site has to reach the target site full of the active agent or of the medicine. And once it reaches the target site, you want the particle to open up and actually spill out that medicine when it reaches the target site. So that's an engineering factor that you have to put into the engineering of these nanoparticles, no matter what you do. And just to put everything onto the same page, these nanoparticles are widely used today in the medicine today. For example, Doxil, which was invented in the Hebrew University, actually in the same lab I did my PhD in a couple of years before I did my PhD. These nanoparticles, which are 100 nanometers in diameter, contain a chemotherapy called doxorubicin inside them and are used today for treating breast cancer and ovarian cancer patients. About 600,000 patients every year use this medicine. And this was approved, this was the first nanotechnology drug to be approved, 1994. And since then, really, the field has been booming. Many, many, many different nanotechnologies are being used for treating patients. And Doxil is injected intravenously to the arm, targets the tumors, but you can also use nanoparticles that are inhaled in order to treat lung disorders, for example, for treating COVID. These nanoparticles were actually adapted and anti-inflammatory drugs have been loaded in. You can load a combination of medicines into the nanoparticles, meaning not only one drug per particle, but multiple drugs per particle. And I think the biggest change in the field is actually the approval at the end of 2018, December 2018, the FDA approved this technology called Onpattro. It's a lipid nanoparticle, about 100 nanometers in diameter, which has, instead of a small molecule drug, actually a RNA drug. So this is the first RNA drug that was approved ever for treatment. And in fact, this RNA drug is not a messenger RNA, as we're well aware of from the COVID vaccines. This is an siRNA, short interference RNA, which instead of for encoding for protein, it actually stops the production of protein. And I'll talk a bit about that technology. But just to frame this, the importance of this this development, when it was invented, it was invented for treating a disease of the liver. This disease is called HATTR, where proteins malfunction in the liver. And from the liver, these proteins settle and actually are distributed to the nervous system of patients and cause a very, very terrible side effects that also eventually lead to patient death. And the siRNA, which was injected to the patients, you can see here, actually improved their quality of life greatly. It stopped the disease. The nanoparticles were injected to the arm of the patients. These are nanoparticles that are engineered to target the liver. The nanoparticles entered the cells of the liver and stopped the production of this diseased protein inside the liver. And after 18 months, which was a very short period of time, the FDA stopped the clinical trial. They said that the clinical trial is so significant, the data is so good, we can't continue. It's unethical to continue this trial, and we should stop it right here and allow the untreated group, which was deteriorating in their therapeutic score, to also get this very important medicine. So that opened the door when COVID actually outbroke to use nanoparticles loaded now not with siRNA, but with messenger RNA inside them in order to treat COVID. And the COVID vaccines are actually based on the same concept of delivering siRNA injected differently, not into the vein, injected to the arm for people who've been vaccinated here. And of course, this is another step, I think, in the evolution of lipid nanoparticles and nanotechnology for delivering RNA. So it's from small molecules to RNA. Just to get a feel, You can see here, these are vaccine particles under an electron microscope, and their size is again around 100 nanometers in diameter. And they actually have the RNA packed into them, allowing the delivery into cells, into most likely dendritic cells, where the spike protein is encoded, and then the patients actually generate an immune response. So to put things into perspective, how small is nano? I always think of this example. This is a small silicon chip, about 1 millimeter on 2 millimeters. And even though it's so small, actually at the Technion, they were able to engrave on this silicon chip the entire Bible in Hebrew with all the inscriptions on it. So they carved using a nano tool, a nano electron microscope, the entire Bible on this small silicon chip. So that really gives it an example of how small nano is. And I always think of it that each nanoparticle is about 1/1000th the width of a hair on our head. In our body, the nanoparticles are about 1,000 times smaller than any cell. And the same also is true for plant cells and also for other types of cells that we're aware of. So a nanoparticle is actually extremely small, and it can deliver an active agent to these cells. So the first example I'd like to show in agriculture is we were looking to use nanoparticles loaded with short interference RNA, siRNA, in order to stop protein production. But this project actually started a couple years earlier when I was working in MIT on delivering siRNA for treating diseases at the time, it was diseases in mice, of course, to develop a therapeutic. And then we made a shift with this technology, changing it for actually for agriculture. So the history is that we use nanotechnology in order to stop the production of proteins inside disease and agricultural disease. I'll show in 1 second. So the science behind it is a discovery at the early 1990s, where researchers, Napoli and co-workers, were using a gene therapy system. They wanted to encode for a color in petunia flowers. They add the gene to the petunia flowers, and instead of getting stronger color, all the color actually disappeared. These are the images from their picture. They didn't explain the phenomena, but in 1998, Fire and Mello explained the phenomena that RNA, short interference, short RNA molecules can actually stop the production of proteins inside cells, and they won the Nobel Prize in 2006 for this major discovery. The discovery actually showed that RNA molecules, which we usually think of them as molecules that encode for the production of a protein, actually can become a control molecule, where the RNA molecule stops the production of protein. And this is a short version of RNA. But when we look at RNA and delivering it into cells, it's actually a huge challenge, because RNA molecules have a negative charge on their surface, and they don't want to penetrate into cells. And that's why you need a delivery system. You need a nanoparticle or a different delivery system in order to carry the RNA into the diseased cells wherever you want to treat. And I think this was best put by a Nobel laureate, Professor Phil Sharp from MIT. He said in the past, and I think it's still true today, the three biggest problems with RNA interference, RNA therapeutics remain delivery, delivery, and delivery. Really, how do you take an RNA molecule, an RNA sequence, and deliver it into a cell? And our approach was actually to take an old molecule that was used in the past called polyethylenamine. The polyethylenamine has amine groups on them which are positively charged, and they can complex together with the RNA, which is negatively charged, in order to deliver it into the cell. However, polyethylenamine alone is somewhat toxic. And what we did is to reduce its toxicity, we added lipid tails to the polyethylenamine through a very simple chemical reaction. And what we created is we created polyethylenamine backbone, the positively charged molecule, and we added lipid tails to the polyethylenamine And we tested its ability of many different variations of this molecule to carry RNA into a cell and knock down the production or the synthesis of a protein. And what we screened about 1,000 compounds, and the successful ones were those that actually were able to stop the production, the synthesis of a protein inside cells. And this is all in cancer. That was in cancer cells. We tested it in additional cells, and then also screened it in mice. And we found a nanoparticle that's formed of these polyethylene molecules and the RNA. Actually, it's all one complex, and that delivers RNA very efficiently for mice. And what we found, and this is actually a very important point, is that while most nanoparticles are taken up very well by cells, some nanoparticles are effective while others are not. And the reason is that the nanoparticle, once it reaches the target site, enters the cell, has to open up and actually release the medicine or the RNA in order to induce the therapeutic activity. And this is what these nanoparticles look like. They look like an onion, where each layer in the onion is this polyethylene material, and the dark regions between one layer and another are the RNA molecules. So the short interference RNA is actually these dark regions inside each one of these nanoparticles, and this is a full nanoparticle, another nanoparticle. And you can see that their size is about 60, 70, 80, 100 nanometers in diameter, give or take. And they were extremely efficient in delivering RNA in mice. We treated different diseases, and unfortunately, this project didn't reach the clinic for certain reasons. And today is actually being evaluated for therapeutic use. And we asked ourselves, we have a very good platform for delivering RNA. Where else could RNA delivery actually be relevant? And when I returned to Israel, I learned about a problem that exists in aquaculture, where shrimp actually are subject to viral diseases, which are every year wiping out more than 20% of the global shrimp production. So this is a virus, it's called white spot syndrome, which affects shrimp and actually prevents from the growth of shrimp or the growing of shrimp in aquaculture farms. Just to show how significant this problem is, you can see that the first time of the outbreak of the white spot syndrome virus, that's what it's called. Until then, from 1976, the growth of the production of shrimp around the world. The first time the virus hits, we're talking about the year 1999, and huge decline in the ability to produce shrimp. Until today, it's been a major challenge. This virus outbreak comes out and actually affects many different regions in the world. Now, I think when I spoke about viral diseases in the past, it was hard to convince people that this was important. But today, there's no question about the importance of treating viral diseases. In general, we've all been subject to COVID, so we understand what a serious viral disease is. And what we did is we tested the ability of a formulation to actually carry RNA into the cells of the shrimp and knock down a gene. And you can see here, we tested different regions inside the shrimp. This is the expression of a protein called RAB7, and we were able to knock down the RAB7 almost throughout the body of the shrimp. And this showed us that we potentially can also knock down the production, the synthesis of proteins, or knock down the RNA that's associated with the viral diseases. So we use this as an example. In fact, we tested the concentration, and we saw that we were able to knock down at different concentrations, and depending on how significant you want the knockdown to be. But an interesting thing about working with RNA in agriculture in general is that longer RNA strands are actually more efficient. So if we look, for example, in mice, 20 base pairs usually are enough to knock down a gene very, very efficiently. Contrarily, in agriculture and in aquaculture, longer RNA strands are more efficient than shorter RNA strands in knocking down a gene. And that's something for people entering the field, I think you should always keep in mind that sometimes you need a longer strand than those that you use in mammalian cells. The knockdown stays in the shrimp for, depending on the formulation, but you get gene knockdown for, initially we got for 14 days. Today we're able to knock down the gene for about 2 months inside. So you get a profile of the gene knockdown, and then it starts recurring, and that's when you need to redose your treatment. And again, this is how efficient your delivery system is. The more efficient the delivery system is, the longer the knockdown and the more efficient. Just from why use a formulation? So in aquaculture, I think it's very clear. In aquaculture, there are many, many enzymes that are out there. And of course, the RNA would be totally degraded if it wouldn't be inside a nanoparticle. And we saw that also outside the shrimp's body, but also inside the shrimp's body, meaning in the digestive tract of the shrimp, RNA is totally degraded and you can't get it in. So you need to have a formulation, a particle, in order to deliver your RNA into the shrimp. From the challenge, though, is actually to get it not by injection, but by food, meaning that the shrimp will eat a pellet, a feed pellet. The RNA will be taken up and then distributed inside the shrimp's body in order to knock down the gene. And that's what we did. We invented a feed that contains the RNA. But first, I'll show an example of RNA particles that were injected to the shrimp. And today we have it already as a feed. So just to show, to emphasize how powerful this treatment is, you can see these two boxes. While animals that are subject to the virus, all the animals die. When you treat them with the siRNA, you actually can get above 90% survival in of the shrimp. And this is what it looks like. You can see this is we ran these trials also in the United States and also in Thailand. And when you treat the shrimp that have a virus, if they don't have your siRNA treatment, all the shrimp die within approximately 1 week. And when you treat them, actually almost all the shrimp survive. So this is extremely, extremely impressive in being able to protect the shrimp. And today I mentioned that we give this by feed. In the interest of time, I'll show 2 additional short examples of using a formulation also for treating diseases in agriculture, and really how agro and nanotechnology can interact one with another. So I showed that the nanoparticle we had before, and I did not elaborate, but the wall of the nanoparticle, in this case it's called the liposome, is a hydrophobic core, and the inner aqueous core is a hydrophilic core. So you can actually load different materials into these nanoparticles, and also combinations of hydrophilic and hydrophobic materials depending on the need. For example, you can add biostimulants or fertilizers or herbicides, and when you come to construct your particles, you want to choose the building materials in order to best address the needs in the field. And the first example is actually foliar application of nanoparticles. So here what we had, tomato plants, we sprayed them with the nanoparticles, and we looked where the nanoparticles actually distribute in the roots of these plants. So we spray the leaf of a tomato plant, and then we look for the nanoparticles in the roots. This is an image of the root after 24 hours. And you can start seeing nanoparticles accumulate in the root, even though it was sprayed in on the leaf. After 48 hours, almost all these cells already have nanoparticles, these green dots that you can see actually inside the root. 72 hours, the nanoparticles start falling apart, and you can see, or opening up, you can see that the dye is actually more distributed. And after 96 hours, the nanoparticles open up, spill up their dye, and actually paint the entire cell. So you have a system here that you can spray leaves and actually go down all the way to the roots of the plant. And when we look at it actually on the leaf itself, we see the uptake of the nanoparticles. These are protoplasts of the cells of the plants, of the tomato plants. And you see very nicely that the nanoparticles actually also reach the nucleus of these protoplasts. We asked ourselves what happens, that we looked initially from the leaves down to the roots, what happens from leaf to leaf. And also there we saw that when you spray one leaf, you get nanoparticles in the other leaves. So you have a transfer mechanism from one leaf to another. And of course, that's very important when you come to treat agriculture, because sometimes when you spray, you actually don't hit all the leaves, and you want to have a system that can distribute to different leaves inside the crop. And an additional experiment we did, we looked at a deficiency in iron deficiency in the tomato plants, and we were able to supplement using nanoparticles. We were able to load them with the ferrum, and the ferrum was taken up extremely well, much better than the free ferrum, meaning that when you have a nanoparticle system, it's taken up very efficiently by the leaves, and then it distributes throughout the plant, so you get a very strong effect on the plants themselves, and are able to actually fix their deficiencies that exist in the crops. When we asked ourselves, what's the kinetics? I mean, when you spray one leaf, how does it reach other areas? So of course, as the farther you are from the application point, depending up or down of the plant, you see the particles, but that changes depending on the distance from the application point. In the interest of time, I'll say that it works also from the roots all the way up to the leaves. So you can spray from the leaves, it goes down to the roots, from the roots up to the leaves, and also between the different leaves. As we were working on this project, we actually tested grapevines. These are beautiful grapevines in central Israel, and we were asking ourselves, can the nanoparticles actually transfer from one region to another in the grapevine? You know that in grapevines, there are regions that are very green, but other areas that actually have a very fibrous tissue to them. And we saw that we were able to actually transfer the nanoparticles from one region to another. The nanoparticles are here immersed, not sprayed. In this case, they were immersed, and we measured them far away inside the plant. And a couple years a couple months, after running this trial, the owner of these grapevines told us that he had a problem in a different grapevine of his, where a viral infection actually affected the grapevines. And he asked us, could we use our formulations for treating the viral disease that he has in his grapevines? And this led to a paper we recently published in Advanced Functional Materials that really looked at the ability to deliver siRNA to grapevines in order to treat viral diseases. Now, if the white spot syndrome, which I mentioned before, is a huge problem, grapevine leaf roll disease is considered to be a true pandemic in grapevines, affects all the blue regions right here. And I actually think that also you guys are aware of this disease in all South America. So what does the disease actually look like? This is a healthy grapevine. You can notice that the green and luscious leaves. On the right-hand side is an infected grapevine, which has a virus called the leaf roll virus. The leaf roll virus changes the color of the grapevines and actually has huge effect on the production of wine from these vines. More specifically, you can notice that a healthy grapevine will look like this, and the berries are smaller, they're discolored when the virus affects them. But most importantly is that the Brix, which is the amount of sugar per grape, is decreased when these viruses occur. And today there's no treatment for this virus, just like with the shrimp. And what we did is first we characterized the virus. We looked for a target at this leaf roll virus that we could use RNA in order to interfere with the virus's ability to infect the grapevine. And then our workflow was this. So first you analyze the disease, then you create a particle that contains the RNA inside of it. And then we go and we treat the grapevines, and we analyze if the virus was actually knocked down inside the grapevine, and we also test for functional parameters in the grapevine. So the first thing we did is actually we synthesized the particles that contain the RNA. These particles are slightly larger than the ones I showed before. They're about 200 nanometers in diameter, but they're very stable over a long period of time, which is very important for agricultural use. You can spray them on the leaves, and they will actually be taken up by the leaf in order to knock down the virus. In fact, when we tested them, we saw that the nanoparticles, again, just like I showed before, these are slightly different. They move also up and down from the application site. And we don't know that they're taken up by the stoma or stomata of the that are in the leaves. This is an electron microscope of the stoma, and you can see that the size is much bigger than the size of the nanoparticles. So this is about 6 micrometers in diameter, over 16 micrometers long, which we have on all the leaves, and the nanoparticles are only 200 nanometers, so significantly smaller than this pore. And then Aviram, the lead author, actually went and sprayed the grapevines, treated them, and tested 2 different applications, one by immersion and one by spray. And he found that there isn't any big difference, but between the 2 application sites. However, the treatment is extremely significant in knocking down the virus. You can see here infected grapevines, very high titer of the virus. And then when we treat using our application, we actually see an almost total eradication, knockdown of the virus inside the grapevines. And more than that, we actually saw that when you treat multiple times, you're able to actually continue the knockdown of the virus inside the grapevine. But more than that, you actually start seeing recovery of the grapevine. So for example, here on the right-hand side of your screen, you can see the Brix level. The Brix is the sugar level inside the grapes. A healthy grapevine has very high sugar levels. A diseased, viral infected grapevine has very low amounts of sugar. And when you start treating with our treatment, you actually see that the sugar actually content starts increasing back to the normal and healthy regions. So this treatment was extremely efficient and made us very happy that we can actually use a technology which wasn't used in the past in order to help the grapevines recover. So I'll end my talk here. My time is up, and I'll just say one great thank you for allowing me to deliver a talk today, and really to end by saying that nanotechnology has a very important role in human health, and I think we're going to start seeing more and more nanotechnologies in the use of animal and crop health. And I think what we need to, one way to approach this is to learn from a lot of work that was done in treating of diseases in human health in order to transfer over and treat diseases that we have in agriculture in order to improve agricultural production. Again, thank you very much. I'm sorry I can't be there in person. I wish I will visit Mexico in the next coming years when this pandemic slightly calms down. I hope we're in a better time. Thank you.
Well, thank you very much. Yeah, thank you very much, Dr. Schroeder. We will be very happy to have you here in Mexico, but I believe if you ask us, most of us will be more happy to visit you over there in Israel, but we can do both.
We can
so,
share, exactly.
Yeah, absolutely. And I think I cannot see any questions. Okay, I will start reading your questions, but also for this session, I will ask people who want to speak up. Yeah, Dr. Riquelme, yeah, had a question before, so I will ask her if possible to open her mic and just go ahead. Thank you. Thank you, Maricel.
Hi, Avi, thank you very much for your talk. This was fantastic. And as Jimena has said, we would love to have you here, but Haifa is also a great city, which I had the fortune to visit. So we'll be very happy to return. I have more of a cell biology question because I was really intrigued by the mechanism. I don't know if it's known, but what is the mechanism by which the nanoparticles, or liposomes in this case, reach the nucleus in the example that you gave, in the protoplast. So my question is, because I work with secretory vesicles, and we know that most secretory vesicles fuse with plasma membranes to reach their target. But in this case, how does a lipid membrane reach the inside of the cell and go through the plasma membrane?
Yeah, I have to say that when we started the field, so people told us several things. One, they said that you can't actually penetrate a leaf because the nanoparticles, that's what we were sure that the cuticula, right, would stop us from penetrating. And then we tested multiple times with many different things, and we saw that the particles are taken up. Same goes for the cells. And I have to say that my feeling, and I'm saying this very carefully, is that a lot of the dogmas we have in the field of plant cells have to be retested again. People in the past used to think that only particles that are 2 to 3 nanometers in diameter can penetrate a cell in a leaf, for example, or in a plant. And if it was only our results, then I would say don't believe us, because maybe we're wrong. But because in the past 2 or 3 years, tens of groups from around the world independently using different types of nanoparticles, iron oxide, gold, lipid nanoparticles, all have shown that you can actually allow the uptake of larger particles. The biological mechanism, and here I agree with you 100%, remains unknown. And we don't know exactly how it happens. We see that it's inside. We tried to capture the moment of the uptake, which would have been a great achievement for us, but we weren't able to do it. We wanted to publish the paper, but I think that's, for example, one of the big questions to be shown. And today it's also possible from a technological perspective, taking a cell, putting a nanoparticle outside, and actually capturing those that event of the uptake, I think is a huge question. What changes in the cell wall to allow the uptake of the nanoparticle? So I totally agree with your question. I feel a bit ashamed that I don't know the answer, but I have to say, if it was only us again,
I don't either.
It would probably be that.
That's
but
fine.
I hope other people actually will be able to also answer that. But I am sure that they are inside. So that I have to say.
Right. No, I think it's also, yeah, I think we all, believe very well, because it's been shown also in the fungal world that they're able to penetrate through the cell wall and they squeeze and change shapes and all that. But yeah, just thinking about the other barrier, the plasma membrane, that's what's really, yeah.
I agree with you 100%.
Okay.
And I think
it's
a big question. Thank you.
Thank you so much.
Okay, Avi, I have some questions from the chat I want to read. Let me see. Ariadne Vegas asks, Dr. Schroeder, how many times you have to treat the plants for the recovery of the vines, the grapevine plants?
Yeah, so I'll say that we treated, and we don't have full recovery. It's important to say, we treated 4 times in order, 4 to 5 times in order to see significant recovery. So we treated every other week. We sprayed every other week in order to strengthen the effect. A very interesting finding we had is that when you treat, let's say, this season, and then you come back next season to the crops, you actually see that crops you treated in the previous season are much better this season. So that's a very recent finding we had when you follow. So you need to give multiple treatments. There's no question about that. It's very important to, again, to use a long RNA. I think that's also very important for people that want to run this trial. But multiple treatments are important, and you'll probably have to calibrate based on what your target is, how many to actually give in order to see the long-term effect. I think like any crop that actually suffered such a viral insult is that it will take time for the crop to actually recover back. But the alternative today in grapevines is terrible. People are actually rooting out the grapevine. So I'm saying even if it takes 2 years to recover or to become better, I think it's worth it.
Yeah, and I'm sorry, I will give you a parenthesis here. We are very aware of GLD. We have GLD over here in Baja California, so we are open to collaborate with you if we want to try your nanoparticles for sure. GLD is a problem over here for sure.
Yeah, so we'll be glad to collaborate and to send the particles over. Absolutely.
That would be great. Thank you. Let me follow these questions. Several questions from Dr. Julissa. So the first one, how do you measure the stability of the nanoparticles in field conditions?
Yeah, so we tested multiple different aspects regarding the stability. So first, stability, which is very important, is actually just in a jar where we've synthesized the nanoparticles, and we test them, room temperature in order to make sure they don't start agglomerating. And that's like the first treatment that we check. We test also for the RNA to make sure that the RNA isn't degraded inside the particles, and that's the second treatment. When we come to spray the nanoparticles, one of the important things to check is that when you spray them, they actually don't fall apart during the spraying process. And so that's some of the field constraints that we actually look at in order to look at stability. And I think you want to see how far the nanoparticle, when it's airborne, is still stable. We saw that 3-4 meters, which is relevant also by using a spraying system, for example, a tractor that goes through the grapevines and sprays them. That's relevant. I will say that an important factor is also when you spray, not only to spray from the top of the leaves, but also from the bottom of the leaves. And of course, choosing an hour. Aviram, who did these studies, used to spray very early in the morning, so about 5 a.m., where the sun starts rising, and that way you also make sure that the uptake is better into the crop. So I think all those factors should be tested in each location in order to make sure that you have not only stability but also uptake. But stability, we tested also different temperatures, many different aspects of spraying and temperatures that would be relevant for agriculture.
Yeah, thank you so much. So the next one is I've been reading that some nanoparticles sort the cells that are bad and accumulate there. Do you think it's common for all kind of nanoparticles? I think she refers specifically for silver nanoparticles.
Yeah, so I think, for example, the nanoparticles, it's true, is a very wide term. And when you use nanotechnology, for example, we chose to use materials that are used also in the food industry. So they're either allowed for feed or food, or soft materials. And really here, one nanoparticle may be different than another. And we chose to use materials that are already used in the agricultural space. It's also important to note to make here that in some countries, for example, nanoparticles that are 100 nanometers or less have a different regulation than nanoparticles that are larger than 100 nanometers. And I think, because the regulators are there, it's important to respect their opinion. And for example, if there's a cutoff of 100 nanometers and you work at 200 nanometers and you still get very good uptake, then you know you can work at 200 and then it's not considered a nanomaterial. I would say that so selecting the right component and at the right size are both very, very important also for the regulatory path, so that you don't have a too long regulatory path, but you still get the activity. If it's a 500 nanometer particle, usually it will not penetrate well into the cells or into the plant. But if it's around 200, it's usually very, very efficient.
Yeah, so we have many questions. I will try to read all of them, but well, the next one is, did you observe nanoparticles in some other tissues, or is it root specific?
So we're talking about, or is it, sorry, I didn't hear the end of the question.
Or is it root specific, or you are able to find in another tissue?
Yeah, so I think the one tissue we haven't looked in, and it's very important, for example, the berries. Do we find the nanoparticles in the berries? That's something that we're gonna go and test. We see the effect of the RNA, for example, in the berries, but we didn't check if we see the nanoparticles in the berry. That's something that we will test for. I think it's important, though, also to choose a material that biodegrade, meaning that after, let's say, a month after the last spraying, you'll see that you won't find the nanoparticles inside. They'll degrade into the building components, and those will be broken down inside the crop. So I would say, though, that they're not root specific. They actually, when you spray the leaves, you see them also in the other leaves, and you see them all the way down to the root. So it's actually, you find it throughout the body of the crop, and not only in the roots. You actually see that it very, very good distribution of the particles. And it makes sense. They enter the different trafficking mechanisms. That's how we see their distribution. I'll say something that's just interesting, that usually you'll see a lot in the leaves and less already in the distribution of the site of the plant, because at that stem petiole connection where the nanoparticles have to exit the leaf and enter into the trafficking modes inside the stem, there's actually that there is a small barrier which doesn't allow all the nanoparticles to penetrate efficiently.
Yeah. All right. So next one is about the resistance of the nanoparticles in the environmental conditions, wind and sun, when you apply on the foliage.
Yeah,
because this is related to your previous answer, right? So
yeah, so I think it's very important to choose the time that you spray and also the size in order to make sure that you have minimal loss and really maximal uptake by the leaves. Extremely important.
Thank you. I will jump over here to my Q&A because I find here some students' questions. So colleagues, please wait a second. Doctor, what is the explanation for the short length of RNAs to knock out in the shrimps specifically?
Yes, so sorry, I didn't say. So in shrimp and in crops, in grapevines, we use actually a long RNA, about 250 base pairs long. In humans or in mice, we use about 21 base pairs. And the thought behind it is that the mechanism is slightly different in humans and mice versus shrimp and crustaceans and grapevines. And what we think, one of the things that happens is that actually in the grapevines and in shrimp, actually the RNA, the long RNA, is cut up into shorter RNA fragments, which are then transferred from one cell to another through the intercellular transfer mechanisms, which then allow better knockdown inside the crops. In humans, we don't have that mechanism, meaning that a nanoparticle, for knockdown to occur, a nanoparticle has to deliver that RNA into a specific cell. It usually is not transferred to another cell directly, unless there may be an exosome that takes some of that and passes it on to another cell. So the transfer mechanism from one cell to another is actually more efficient in agriculture, and that's why we use a longer strand. By the way, the nice thing about it is that you can even hit a small number of cells, and then you get distribution that's much more efficient.
Thank you. Another question from our student, Rommel. What are the main challenges for nano agro biotechnology to go from lab to the market and for its general use in the crops?
Yeah, great question. I think that one challenge that always exists is scale up in pricing. You have to, in agriculture, at the end of the day, you have to be competitive in your price. And I think one way to approach this is to choose a problem that doesn't have any other solution. For example, the viral diseases in shrimp have no other solution. Leaf roll virus, to the best of my knowledge, doesn't have a very good solution out there that's good for treating the grapevines. And so one, it's a high quality crop, right? Grapes, which are more that. And two, there's no solution. So I think that's a good place to be. Another aspect which has to be considered is actually the regulatory side. And you want to ask yourself that in the beginning. I mean, what questions will we get from the regulators and how can we engineer our particles so that they're safe from the regulator's perspective? I think once you cross these two boxes, one, having being able to solve a real problem in a cost-effective method, and two, not having too many challenges from the regulator, your path is pretty is a good path. So choosing the problem, I think, is very, very important.
All right. So I think we still have some time. So another question from Dr. Perez Almeida. So secondly, the technology is wonderful for grapevine viruses treatment. Do you think it will be useful in the treatment of Fusarium race 4 in bananas? Have you thought about making some research on this area? It's very important for banana producer countries. Yeah.
Yeah, so we've thought of it. We haven't tested it yet, but we've thought of it. I think it's a very sensible approach to go. And we started with grapevines, one, because my colleague, Professor Oded Shosayov, was aware of, he grows a lot of grapevines, and he was aware of this disease. So we worked together with him on this. I think bananas surely are also another target.
All right. Let's see, let me jump over here to the chat. What is the reason of the accumulation of nanoparticles in the root?
Yeah, so what we see is actually, we see transfer also from the leaves down to the roots and also from the roots up to the leaves. So what we assume is that through the liquid mechanism or liquid transfer mechanisms that we have inside plants, the nanoparticles actually traffic in those up and down the plant in order to reach. I know the word in Hebrew. I'm trying to think of it in English. We call it etzah and shifa. One is actually transfer up, and one is transfer down inside the plant. In the nanoparticles, most likely that's how they transfer throughout the crop. It's in the stem itself. You have two regions, one taking up, one taking down, and that's how probably the nanoparticles are trafficking.
Thank you. I think this one, the optimal concentration of the nanoparticles that should be used in real situation, I believe you already tackled that one, but I think it's the last one. So if you want to comment anything. Oh, excuse me. No, we have another question from Dr. Kurlango. So Gil, if you want to open your mic, please go ahead.
Thank you for your talk. It was very interesting. I just wanted to know if you have any input or any information to share about the delivery of DNases using this technology to treat cancer cells.
The delivery of DNAs, for example?
DNases, enzymes.
Yeah, so for the delivery of, yeah, I have something here with my line. So the delivery of DNA?
DNases.
Oh, okay. So in cancer cells, the delivery of DNAs, yes?
Delivery of enzymes like DNases.
Yes, so recently, yeah, sorry, I had here, my internet was fluxing. So we published two papers in 2018 and 2019 on the delivery of collagenase, not of DNAs, but an enzyme that's an anti-collagen enzyme. for treating pancreatic cancer. And in pancreatic cancer, there's a high expression of collagen. And we loaded an enzyme, collagenase, which is specific to collagen, into liposomes. In this case, about 100 nanometer liposomes. We loaded the collagenase internally. It's very important when you load an enzyme to make sure that the enzyme is not activated when you load it, because each enzyme has a time of activity. And if you load it and it's already active, then usually once it reaches the target site inside the body, it's already inactive. So you want to make sure that you use the right salt concentrations to load it in an inactive form. And then when it reaches the target site, it opens up, and then the biological environment actually will activate the enzyme, and then it will be active. So I'm aware of people that have delivered. There's even an Israeli company that worked on delivering of DNAs for treating different cancers, including pancreatic cancer. And it makes a lot of sense to me, because there's a phenomena of really extracellular DNA in different types of cancer. It's secreted by neutrophils, which are cells of the immune system, and creates nets actually in the site of different cancers. So even though we haven't delivered DNAs, we delivered collagenase for a similar approach. So I would say that that approach is totally makes a lot of sense, and has a therapeutic potential.
Thank you.
Thank you.
Okay. Well, I just want to say thank you again for sharing with us your time. I believe it's like 8 p.m. over there,
Yeah,
right?
yeah, exactly 8
So
p.m. Time for dinner.
thank you so much. Good night. And we will be more than happy to have you here whenever it's possible. And
thank you very much, and looking forward to seeing you in person, either in Israel really or in Mexico. Thank you,
there
everyone.
are several and very, very happy people around here. Congrats, thanks, and everything. You can see the chat. Thank you so much.
Thank you. I appreciate that. Bye-bye, everyone.
Bye.